Hey everybody, this is a lesson on DNA transcription and translation. I call it From DNA to Traits. So we're going to start off by taking a look inside of the cell. You might recognize this. We have our nucleus right here. Outside of the nucleus is the cytoplasm, so you can't see the whole cell. We're zoomed in. We are going to start off by focusing on transcription. So transcription is the first step to making DNA into actual stuff. DNA are the instructions. The entire instruction manual is in the nucleus of each one of your cells. And we have to take those instructions and turn it into real things. And those real things that we make are proteins. Proteins carry out all the jobs inside of your cell. So we're starting with transcription. That is happening in the nucleus. Here is our DNA. You're going to recognize that double helix shape. And if we zoom in, oh, not zoomed in yet. Here's our definition. Transcription. When you use DNA as a template to produce RNA molecules. You might be thinking, what's RNA? Well, RNA is a different kind of nucleic acid. That's what the NA is for. And it is a code just like DNA, and it is instructions as well, but it's just a little bit different, and it's kind of like a copy. It's kind of like if you go to the copy machine and you take a chapter from a book and you make a copy of it. That's what RNA is. It's the copy of the instruction manual. So this RNA then travels outside of the nucleus, which is also important. We have to take the information and get it outside of the nucleus. So the DNA, since that's going to be in the nucleus, we're going to make a copy using RNA and move it out of the nucleus. Because then in the cytoplasm, that's where our ribosomes are, and the ribosomes can turn that information into actual proteins, which are going to do the real jobs in the cell. So the process of making that copy of RNA is called transcription. So transcription, once again, we are in the nucleus. It's in the nucleus. We're making a copy right here. Let's zoom in. You can see that you've got your DNA double helix here, and there's this giant blob that has latched onto the DNA. That is a protein that helps to unzip the DNA. Remember, it looks kind of like a zipper. And that's DNA polymerase. That's the fancy name for it. And it's going to open up the DNA and reveal the code here so that we can make a copy. You can see this blue line right there. I just colored it black. That's the copy that we're making. So you only have, genes, they code for all kinds of stuff in your DNA. And different sections of your DNA code for different kind of traits. So you don't need all of the instructions all at once. You just need little sections. So you can make little copies of the section you need. So the blue, if you look at this bottom picture, this actually has the base pairs, the code visible. The top one doesn't have the code, it just is colors. But you can see the different letters representing the different bases: adenine, thymine, cytosine, and guanine. Now the DNA, you see A, T's, G's, and C's, those look familiar. That's what our code is written in. But if you look at the mRNA, and you might be going, mRNA, I thought it was just called RNA. Well, there's different kinds of RNA. This one, the copy that we're making inside the nucleus, we call mRNA, which is short for messenger RNA, because that's the one that carries the message from the nucleus to the cytoplasm. So that RNA is called messenger RNA. You might notice that right here we've got a U. We've never seen a U before. U is in place of T. So when you have mRNA, you don't have any T's. All of the T's get replaced with U's, and U is just a different kind of base. It's called uracil. And that's one way that you can tell that it's mRNA. It's a copy, because instead of T's, we've replaced them with U's. Okay? So we are making a copy of the section of DNA that we want, and we're making it, that copy, in mRNA. Then that copy, that mRNA, it goes through a little bit of a proofreading process here. Then it goes out of the nucleus into the cytoplasm. So our mRNA is traveling out. It's carrying the message out of the cell. Now, the next step, so that was transcription. That was it. We just made a copy of the section of DNA we wanted, and that piece is called mRNA, but the whole process of making the copy is transcription. You're transcribing a copy. So that was it. That was transcription. Now, when we take that mRNA and we go into the cytoplasm and we're going to do our next step, that next step is translation. So we've made a copy. Now we're going to translate that mRNA copy into an actual protein. So translation is where we get to make proteins. Yay, we want proteins. Those do the jobs. All right, this thing right here, that's a ribosome. Ribosomes make protein. So let's figure out how a ribosome is going to make a protein out of mRNA. So translation, what's the definition? Using RNA, specifically mRNA, messenger RNA, to assemble amino acids into a protein. So a translation, we're taking RNA, reading off that copy, and using those instructions to assemble amino acids. Those are the building blocks of proteins. We put the amino acids in order, stick them together, and they form a protein. So let's look and see how that happens. We are zoomed in here. This is a ribosome. Ribosome actually, a ribosome is actually two subunits put together. So there's a top piece and a bottom piece. And they attach onto this mRNA. The mRNA moves through the ribosome. And the ribosome actually has more RNA to help it turn the code into an actual protein up here. So the thing that's helping it out are these big yellow squiggly things. These are called tRNA. tRNA stands for transfer RNA. This type of RNA is this piece of RNA, this windy, crazy looking thing. It carries amino acids. And each tRNA carries a specific type of amino acid depending on what the code at the bottom of the tRNA is. So you can see that these match up to specific spots in the mRNA. If the tRNA matches the code in the mRNA, it'll lock into place. And this helps the ribosome know which one of these amino acids goes where. So the code in the mRNA helps the tRNA, transfer RNA, line up and match up in the correct order so that the correct amino acids get placed into the chain and the correct protein gets made. So there's actually one more type of RNA involved here. You're looking at it, but it's not labeled. The next type of RNA is actually the ribosome itself. This ribosome is made out of rRNA. The ribosome is made out of ribosomal RNA. So that's another kind of RNA that's involved. This is a different picture of both processes. You've got transcription. We are transcribing a copy here of the code from the DNA. So this RNA travels out. It's actually called mRNA, but it's transported out into the cytoplasm. So that's transcription. Then once it's out in the cytoplasm, it's going to find a ribosome. That ribosome is going to carry the mRNA through it, and it's actually the ribosome is going to help these transfer RNAs to attach and assemble the protein. And the order and the type of amino acids in that chain is determined by this code, and the transfer RNAs know which order to go in based on what the mRNA says. So each one has its own identifying set here. This is actually the sets of 3 base pairs are actually called codons. So it reads them in sets of 3. So if you remember from class when I showed you the decoding chart, it was all in sets of 3 letters. That's because those tRNA read the mRNA in sets of 3. So let's review all of the different nucleic acids that were involved in these 2 processes. We've got DNA, that's the main instruction manual kept in the nucleus. We have mRNA, that is the copy. Those are the copies that you make of certain segments of DNA, which travel out of the nucleus, and then those get translated in the cytoplasm by the ribosomes. So messenger RNA, that carries the message, the copy. tRNA, transfer RNA, that RNA is actually carrying specific amino acids to the ribosome, and it attaches according to the code, and that's how the ingredients get put in the right order, in the right place. They transfer it there. They transfer it there, and the ribosome actually assembles it, helps it attach all together. And the ribosome is made out of RNA as well, called ribosomal RNA or rRNA. So those are all of the nucleic acids. Nucleic acids are making it happen, for sure. Here's a picture I like because it has all the RNA labeled. It doesn't have DNA labeled. You can't even see in the nucleus of this one. But I like how it has all those different kinds of RNA labeled. You've got your mRNA, messenger RNA with the code. You've got your RNA, which is that ribosome. You've got your tRNA, the transfer RNA, which is carrying those important amino acids and attaching. This is the anticodon. It's the opposite of the codon. And then we know which amino acid goes where. They all attach and end up making a protein. All right, so I would like you to watch the video that's attached with this assignment and then continue with the instructions on the assignment and turn it in to Google Classroom when you are done. Please watch it as many times as you need to so that you can understand the process. And if you have questions, feel free to email me or ask tomorrow in class. Thank you. Bye.